I'm Chloe Robbins. I'm a fifth-year graduate student in the Population Biology, Ecology, and Evolution graduate program at Emory University. And I'm going to be talking about using DNA methylation data to test evolutionary models of human aging. Okay, so aging is defined as the progressive and general deterioration of an organism resulting in decreased fertility and increased risk of death. Aging, then, by definition, causes decreases in Darwinian fitness. And basic evolutionary theory suggests that such a trait should be selected against, yet all humans age. So aging poses an evolutionary paradox and raises questions like, why hasn't aging been effectively selected against, and why does aging exist? Okay, so evolutionary theories provide possible explanations for the existence of aging. Major theories can be classified into two groups, adaptive and non-adaptive. Adaptive theories suggest that aging is a product of evolutionary optimization, while non-adaptive theories suggest that aging is a byproduct of limitations of selection. Okay, so Kirkwood's disposable soma is an adaptive theory. This theory suggests that aging is caused by somatic damages that accumulate over the lifespan of an individual due to imperfect maintenance and repair mechanisms. Medawar's mutation accumulation is a non-adaptive theory. This theory suggests that aging is caused by deleterious germline mutations with effects confined to only late in life that accumulate in the population over time due to a decline in the strength of selection with age. Okay, so these theories are not mutually exclusive, and it's likely that both play some role in explaining the features and existence of human aging. So my work aims to answer the question of what is the relative contribution that these theories make in explaining aging. Okay, so mutation accumulation and disposable soma make contrasting predictions about the heritability of aging-related traits. Mutation accumulation predicts that the heritability of lifespan and other features of aging will increase with age. This is because genes with effects confined to only late in life can experience only weak selection, so that causes mutation frequencies to increase with age, which relates to genetic variation increasing with age, and as genetic variation increases with age, so does heritability. So if you were thinking of some quantitative aging-related trait, and you're looking at comparing the phenotype of relatives, such as twins, you'd expect that the correlations between the relatives would increase with age, or that their phenotypes would converge. So with disposable soma, it's the opposite. Disposable soma predicts the heritability of aging-related traits to decrease with age. Disposable soma predicts an inherently stochastic process of aging because the damages that accumulate in the somatic cells are expected to result from random failure events in maintenance and repair processes. So because of the stochasticity, you'd expect environmental variance to increase with age, which relates to a decrease in heritability. So if you're thinking of a quantitative aging-related trait and the correlations between relatives, you'd see that the correlations decrease with age or the phenotypes diverge. Okay, so to test these contrasting predictions, I used DNA methylation. DNA methylation is an epigenetic modification. It's an alteration that doesn't change sequence. It's the addition of a methyl group to a single nucleotide and typically occurs at CPG sites where a cytosine base sits directly next to a guanine base. It is often associated with gene expression silencing, and it can be measured at over 450,000 sites across the genome. And these measurements range from 0 to 1 and represent the proportion of DNA strands that are methylated at any particular site. Okay, so I'm going to be using DNA methylation as a biomarker of aging. A growing body of literature reports distinct genome-wide changes in the DNA methylation with age. That is, at numerous sites, you can see consistent variation between younger and older ages. You can also use the DNA methylation at just a few hundred CPG sites to accurately predict age across a range of cell and tissue types. And these correlations between chronological age and methylation estimated age are often much higher than other biomarkers of aging, such as telomere length. And then finally, DNA methylation-derived estimates of aging predict mortality better than chronological age. So altogether, this evidence suggests that DNA methylation is an appropriate biomarker of aging and can be used to test evolutionary theories of aging. Yeah? Okay.
Okay, so depends what you want.
Okay. Okay. So DNA methylation is also a genetically heritable trait. So on average, CPG sites across the genome without underlying SNPs show moderate heritability of methylation. So that means that at these sites, the measured level of DNA methylation is more similar between relatives than unrelated individuals. Okay, so I showed you heritability predictions of mutation accumulation and disposable sona before, and I want to test them using DNA methylation. So to do that, I'm first going to extend the predictions I showed you before specifically to DNA methylation. So like I said before, mutation accumulation predicts the heritability of aging-related traits to increase with age, and disposable soma predicts the heritability of aging-related traits to decrease. Sorry, mutation accumulation increase, disposable soma decrease. And I've color-coded everything throughout this. Mutation accumulation, red, disposable soma, blue. So hopefully that will help. Okay, so my underlying assumption is that aging-associated DNA methylation changes reflect aspects of the biological aging process. So, okay, if age-associated methylation changes are consistent with mutation accumulation, they will mediate the age-specific effects of deleterious genetic mutations, and the heritability of DNA methylation will increase with age. If instead, age-associated methylation changes are consistent with disposable soma, they will result from stochastic failures in maintenance and repair mechanisms, and the heritability of DNA methylation will decrease with age. Okay. So the data I used to test this was from the Brisbane Systems Genetics Study. I had DNA methylation data from 610 individuals from 176 families. And these families consisted of MZ twins, DZ twins, their siblings, and their parents. All individuals were of European descent. The ages ranged from 10 to 75, with mean age around 21, median around 14. And then DNA methylation was measured from whole blood using 450 Ks arrays. So we had about 450,000 CpG sites across the genome to test these predictions. Okay, so to test whether the heritability of DNA methylation is consistent with mutation accumulation, or that is, the heritability of DNA methylation increases or decreases with age, at each CpG site, we modeled methylation as a function of age. So XIT describes the methylation of individual I at age t, and then beta age describes the change in methylation with age. So then we modeled the covariance between relatives such that genetic variance and environmental variance could be partitioned into terms describing the baseline and the change in variance with age. So these gamma G and gamma E terms describe the change in variance with age and are what we will use to categorize sites as consistent with disposable soma and mutation accumulation. Yeah?
Could you explain the subscripts, the I?
Oh, I is individual I at age t.
At age T?
Age t, yeah. Yeah, so the T's next to the gamma G and gamma E are also age.
Like longitudinal data?
It's familial, cross-sectional familial. So it's twins, their siblings, and their parents. Yes. Okay, so then we added restrictions to this general model to test specific hypotheses via likelihood ratio tests. So the first thing we tested was whether age is a predictor of methylation level, or that CpG site is age differentially methylated. We then tested whether the methylation level has a heritable component. And then after that, we restricted to sites that were heritable and tested whether the heritability of methylation changes with age by restricting the gamma G and gamma E. Okay, so together these results will allow us to categorize the CpG sites across the genome as consistent with either mutation accumulation or disposable soma, and that will allow us to get at the relative importance of each. Okay, so first I looked at whether age is a predictor of methylation level, or which sites are age differentially methylated. So this is a bidirectional Manhattan plot. Each site is a CpG, or each dot is a CpG site. Along the x-axis is the location in the genome, y-axis is the sign and magnitude of the change in methylation with age. So all the dark green dots have significant increases in methylation with age, and all the light green dots have significant decreases in methylation with age. And sorry, the dotted line is genome-wide significance. So everything colored is above genome-wide significance.
So here you contrast age to methylation level, but you speak about mutation. So did you draw any connection between age and mutation, not methylation?
No, not yet. Just here right now, I'm just looking for sites that show changes in methylation with age.
Yeah, but the work is about correlation between mutation level and.
So I'm trying to test two different evolutionary theories of aging, which make differing predictions about the heritability of aging-related traits. So I'm using DNA methylation as an aging-related trait to test these contrasting predictions between mutation accumulation and disposal-soma. So first, to do that, I'm going to find which sites show changes in methylation with age. Then I'm going to find sites that show heritable methylation levels. And then I'm going to use both those sites to find sites that show changes in the heritability of methylation with age. So let's see if I can. I'm looking for sites that, okay, so I'm looking for sites that either the phenotypes converge with age or diverge, basically, if that makes sense. Yeah. Okay. Sorry.
Phenotype in your case is
Methylation. Yes, DNA methylation level. Okay. So I found around 92,000 CpG sites that had significant changes in methylation with age after multiple test correction. That was about 24% of the sites tested. And 47% of those showed significant increases in methylation with age, and 53% showed significant decreases in methylation with age. And then I looked at the heritability of methylation level, and I found the mean heritability across all sites for DNA methylation level was 0.17, and when then I restricted to just age differentially methylated sites, it went up to 0.2. I estimated a heritability of 0 for about 20% of the sites, and that means after multiple test correction, I had about 37% of the sites that I tested showed significant non-zero heritability. That's around 144,000. Okay, so then I tested, I restricted to sites that were both age differentially methylated and heritable, and then wanted to test whether the heritability of methylation level changes with age, which is what will allow me to categorize sites as consistent with mutation accumulation or disposal-soma. So mutation accumulation, I'm looking for sites that have increases in genetic variance with age, which relates to an increase in heritability. Disposal-soma, I'm looking for sites that have increases in environmental variance with age, which relates to a decrease in heritability. So I looked at the changes in environmental variance and genetic variance separately first. And I looked at the change in genetic variance with age first. And again, this is a bidirectional Manhattan plot. Each site represents a CpG site. The sites that are above genome-wide significance are colored, and they're colored to match the theory that they're potentially consistent with. Okay, so I found 102 sites with increasing genetic variance with age that was potentially consistent with mutation accumulation, 513 sites with decreasing genetic variance with age, potentially consistent with disposal-soma. Then I looked at the change in environmental variance with age. I found 2,266 sites with increasing environmental variance with age, potentially consistent with disposal-soma, and 168 sites with decreasing environmental variance with age, potentially consistent with mutation accumulation. Okay, so I did this, and then to be sure of the direction of change in heritability at any one site, you have to look at the change in genetic variance along with the change in environmental variance. So I did that next. Okay, so this plot is the sign and magnitude of the change in environmental variance against the change in genetic variance. Yes, sign of significance, yes. Oh, yes, okay. Okay, so I found 102 sites that I had significant increasing genetic variance that was greater than the change in environmental variance. So I'm confident that these have increasing heritability with age and are consistent with mutation accumulation. Then I found 2,266 sites that have significant increases in environmental variance with age that are greater than the changes in genetic variance with age, which relates to a decrease in heritability. So I'm confident that these are consistent with disposal-soma. Okay, so in summary, I found around 92,000 sites that showed changes in methylation with age, around 144,000 sites that have heritable methylation levels, and then I found around 3,000 sites where the heritability of methylation level changes with age. Of these sites, I found 102 sites that I'm confident are consistent with mutation accumulation, where the heritability of methylation level is increasing with age, and around 2,266 sites that I'm confident are consistent with disposal-soma, where the heritability of methylation is decreasing with age. So you could think of this as the 2,266 sites that are consistent with disposal you see those phenotypes diverge between twins as they age, and the 102, you see them converge as they age. So based on this, I can say that both mutation accumulation and disposal explain the aging-related changes we observe in DNA methylation, but disposal explains a larger proportion of the aging-related DNA methylation changes when compared to mutation accumulation. Okay, so now I found 102 sites that are consistent with mutation accumulation and 2,260 sites that are consistent with disposal the next thing I want to do, since mutation accumulation predicts that there are age-specific genes, I'm hoping to see if the DNA methylation changes at the sites consistent with mutation accumulation associate with any expression changes. And then for the sites consistent with disposal I'm hoping to test whether or not these methylation changes are truly random. So I'm hoping to get longitudinal data and see if they actually fit a stochastic model. Okay. Oh, yeah. Thank you to all these people. Karen Keneally is my advisor. And thank you. Yeah, that's it.